So with that, we can move on to this notebook. And I hope you guys can access it. So let me know if you guys are able to access it or unable to access it. In this particular example, we're going to be looking at performing a machine learning problem using the Harmonizome. I don't know if it was introduced before, but the Harmonizome is a platform that has a wide variety of data sets across transcriptomics, let's see, genomics, disease and phenotype association. So some of it is literature curated, some of it is actually measured from a lab and then processed into data that you can use. And it's, it's all here. So there's also an API, and the data sets there are downloadable. So this notebook, we're just going to use scikit-learn and actually try and do gene function prediction, using some of the data on this site.
So maybe you should look at who has access and whether you're able to execute or copy this notebook in Colab or locally.
Yeah, I got one thumbs up. Were there others? Yeah, 2, 3, 4, 5. Yes, I think they got it. And what you can do, you can just run it, and I'll kind of walk through with you. And definitely stop me if you have questions about any aspect of this. But these data sets that I chose are somewhat arbitrary, but they, so you can look through the Harmonizome and find other data sets with data that you're interested in, and you could actually test the same approach on your own, like selection of data. And yeah. So the first thing we have to do is install some dependencies and import them. The main dependencies are scikit-learn, pandas, and NumPy, and then Plotly and SuperVen and Matplotlib. So these are for plotting. This is scikit-learn, that resource that I mentioned with lots of machine learning tools, pandas and NumPy for numerical operations. So hopefully that runs. It worked for me. So the first data set that we're looking at is this diseases, which has curated gene disease association evidence scores. This comes from the Jensen lab, and it's been processed by the Harmonizome. And here's kind of the citation for that. And I loaded this straight off of the Harmonizome link that you get in the download section. So if I just run this, this downloads the data set and just loads it in as a pandas data frame, so we can see what it looks like. So this data set has genes here on the rows, a gene name, and then some ensemble name, or rather these are proteins. And then we have on the columns, we have these curated disease names. So a whole bunch of different diseases. And we also have these disease codes. And ultimately, we have a lot of zeros. But if we look into it, we will find that there are some ones. And a 1 would mean, as if in this case, that would mean that this particular gene or protein is associated with this particular disease. So that's how this data set works. So since this is curated from text mining, it's not really going to be that good for actually using for data, because we're just going to be kind of rehashing things we already knew, and things in text and literature are typically very biased. You know, so what we can use this for are labels. So these, for any given disease, we know some genes that are associated with that disease. And that's what we're going to use this data set for. And that's why I called it labels. The next data set we want to use is geosignatures of differentially expressed genes for gene perturbations. So a gene perturbation is performed. So a gene is knocked out, overexpressed, or mutated. And then we measure the effect of that. We measure that effect to other genes in some condition. So in this particular matrix, which we again get straight off of Harmonizome, we can see the data again. It's also pretty sparse, like the other one. We have these various genes, and then we have a number of experiments that were performed. So in this particular experiment, this particular gene was knocked out in a mouse. This other gene was knocked out in a mouse in some different condition. So that's what this data set looks like. And so the theory, if you will, is that we can take this data, which has some intricate kind of associations between genes, because if you knock out one gene and it has this effect on other genes, you're saying that this gene somehow is directly related to that other gene downstream by some mechanism. So we're going to hope that this amount of information, it will be enough to tell us something about a disease. So if we know that this gene is implicated in a particular disease, based on how this gene is knocked out and how other genes behave when they're knocked out, we can kind of infer that these genes somehow have to do with the same disease. So that's what we're doing here. And again, we could use different diseases. We could use a different data set for labels, and we could use different data. But this is what we have. So this is what we'll use for now. So did I run this one? So I'll run it. Then we're going to take this labels, and then we'll just kind of look at the sum, and we'll sort that. And what that's doing is it's just showing me in that particular data set, what are the columns in it. We can see that it's somewhat hierarchical, this data set, because we can tell that disease is here and it has the most entries, and then we have disease of anatomical entity. Presumably, this is a subset of this, and down the line. But each one of these has this many genes that have been shown to be associated. So, we could predict a disease, right? So based on knockouts, or, how can we say if this gene was knocked out and it was implicated in any disease, will this other gene be implicated in any disease? But that probably would not be that useful. So we move down a little bit. We do have a trade-off here where we don't want to have too few samples, or it becomes very difficult to get good results. But basically, for this demonstration, we picked this eye disease. And again, if you like, you can even try to put a different disease in here. and see what results you get. But for the purposes of this demonstration, that's what we picked. So eye diseases, so I put in the name and then I just filter out the labels, and you can see that we get this eye disease and the eye adnexa disease. Because these are probably supersets, it shouldn't matter that much. I can just combine these. So with that run, now we need to kind of prepare our data. So before, this data had these like 3 columns or 3 segments in the columns and in the index, and that's just a little hard to work with. So that's what this code does. It just collapses it down. So now I just have the gene by some experiment. You know, there may be the same gene may have been knocked down in different experiments. We've just left them all in columns. So that will be like a redundant column, but you know, such is the nature. Even if it were a different gene that was knocked down, there's likely redundant information there. So it's simple enough to just concatenate it all. So our data looks like this. And we have now 22,000 genes that we have these experiments. We have like this evidence of being knocked down or knockout in connection with this gene across these different experiments in Geo. So that's what we have. Now the goal really is to first kind of get a sense of what our data looks like. So remember, I mentioned that the first phase of any problem should be the exploration phase. So we can apply this PCA, a unsupervised method, to the data and see what we have. And one thing you'll see about this data is that we have these kinds of, first of all, there's very little shared variance. And second of all, we have these data sets that are very far. And if we zoom in and hover over some of the points, we can see that the ones that are very close together are from the same study, which is to be expected. But as we can see, like across different studies, it could be universes apart. So this is just something to be aware of. We have these like clumps of studies that are together, but in the grand scheme of studies, there's also some studies that are pretty far from everything else. But because the explained variance is so small, it really just means there's actually a lot of like non-collinearity information here. So there's not really anything we necessarily have to do about this right now. But another way to look at this besides PCA, which is clearly difficult because we have such little coverage on the axes, is t-SNE. And as mentioned in the lecture and in previous lectures, the goal of t-SNE and UMAP are to preserve neighborhoods in points in the high dimensional space when you project it into lower dimensional space. And when we do this, again, we can see that points from the same study, in this particular case, we have actually a different study, but we have the same gene that was this HEC293, or maybe that's a cell line.
Yeah, that's a cell.
Yeah. But you know, there's some similarities. They're clustering by some shared thing, but you do have a lot of them. And so this is telling us a similar picture to the PCA, that we just have like a lot of completely separate pieces, small pieces of very diverse information in this data set, which is somewhat promising actually for what we want to use it for, because presumably across all these features, we can we can separate our eye disease points. Okay, so the next thing we want to do, just from a practical standpoint, is we had 2,000 genes that we have disease annotations for, 200 of which are eye disease and a bunch more which aren't. And then we have our data set, which has, 22,000 genes. So this super Venn diagram is kind of like a Venn diagram, but it shows boxes instead of circles. And we can see this is the genes in the gene perturbagens. This one is from diseases, the 2,200. And then we have the ones associated with eye diseases. And then we can see the overlap. So we have 228 genes that exist in both. We have 3 that are only in the diseases and not in our geo. And we have another 17 that are just not in geo and only in the diseases. So we could try and map these genes, but since it's so few for now, I've decided to throw them out. And to do that, there's this align function. And what align will do is make sure that X and Y's axes are the same. And anywhere there's an NaN, I'm going to fill it with a 0. So our Y's now has the same dimensions. So we're just putting in a 0 because it's not known. Before it was still not known, so we're just adding more zeros to this labels. And we have our data where we have all the original genes, basically. So the only thing we threw out were these 3 known labels. But, we don't know those genes, and in the grand scheme of things, it's pretty small. So now that we have our data frame with the axes aligned, we can now actually start to do our cross-validation. So again, we want to make sure that some amount of the data is kept out so that we don't use that to train the model. And that's what this train test split function does. It takes 20% of the data. It's stratified. So this is taking 20% of the eye samples. So we had 200, so it'll be 40 samples or so that are in there. And then we want to actually figure out from the training data that we're going to be using, what is the class distribution. So I just run some code here and we can see that we have this 17,000 zeros and 182 ones. So these are the samples that we know are implicated in eye diseases. And so we can create a fraction out of those. And these will be the sample weights. And we're doing this because our labels are imbalanced. There's a lot fewer zeros than ones, but these zeros are not true negatives. They're actually just like unknowns more than a true negative. And so some way we can counteract and make sure the model is not biased and just says everything is 0 is we can counteract that bias by assigning these class weights. So now, even if you get a 1, if you get a 1 accurately, you get like more points, if you will, than if you get a 0 accurately. And this will, to the model's eyes, it will be a 50-50 split, but the outcome will be kind of better for us. Then I have this function, and all this function does is it fits the data, and then it makes predictions, and then it draws some plots for us. So that will be helpful. And the next thing we want to do is actually apply this model. So The first model we'll apply is a logistic regression classifier. It can be confusing because of the regression in its name, but what it means is it's actually a logistic regression model. That is, it's trying to solve, y equals mx plus b, your classic formula. But what you do is you use this special logistic function, and you wrap the mx plus b part in that function. And what that does is it maps from the real domain, -infinity to infinity, into 0 and 1. So even though you're predicting, the linear model thinks it's predicting something from -infinity to infinity, this regression problem, the actual outcome is going to be a 0 or 1 probability distribution. So you got a classifier on your hands. So this is a neat trick, and you can actually apply this to any regression model to create a classifier out of it. So when we do this with our classifier and we run this benchmark classifier, this particular model has this CV. So remember I showed this cross-validation with 5 folds. So internally, it actually just does that. That's what this CV is 5 is telling it. And this random state is just because there is some determinism, because when we do these splits, we shuffle first. We want to have the same shuffles so that if we run this tomorrow, we get the same result. And so it's typically a good idea to have a random state variable if you're making a paper, because you'll make your plot, and then, if you want to tweak the plot a bit and you didn't do this, you're never going to be able to make the same plot again. So just word to the wise. I should have just left it. It takes a little while. It doesn't take that long, but it does take a little while to run. Oh, there it goes. So once that ran, we can see the first part is the training data. So we gave the model this logistic regression classifier some data to train with, and then we test it on that same data. And that's what this curve is showing us. So what we can see is it's spectacular. It does great. It can just draw a line, and it can do this prediction very well. Here is that grid. So we have this was a true positive, a true negative, and so it got a few wrong, but it's great, right? Now, but the problem is that it already saw this data, so of course it can separate it. When we look at the test data, the data that we never gave to the model, we can see that it actually does worse than random. It got a few of them, but in general, it's not doing great. So this is essentially the issue with every machine learning problem. If you give it data, then it's gonna be able to categorize that data very well, but it does not mean it's gonna generalize well. So there we have our linear model does not generalize, even though it can learn it. And why can it learn it so well? It's just because the dimensionality is so high. This is a very common thing. So let's try something else without going too deep. We can try a random forest classifier. Remember I mentioned that these decision trees are much more powerful than lines. Well, our line can already memorize, so of course a decision tree, a random forest, can memorize perfectly on the training data. And we can see the evidence of that in our ROC curves. But yet again, our decision tree is not that great at generalizing. In fact, it can't learn anything with these default parameters. So, once again, if you see great results, be skeptical. Okay, last thing we'll do is instead of digging into these algorithms more and understanding how to make them do better. So with the random forest, you can tweak these hyperparameters and you can certainly get it to generalize better. But the defaults are they give it so many models. So the random forest classifier has like 100 decision trees by default. Yeah, see 100 estimators, and there's only 200 labels. So each estimator can learn one of them. That's why it's perfect. But you can lower that and it'll do a lot better at generalization. But I'll just end with this XGBoost. So this is used in Kaggle competitions, which is this data science like competition platform. And XGBoost does amazing things. And you can find out more information on their site. But I just threw XGBoost at this problem, and we can see that it doesn't do 100% on the training data, and that's actually a decent sign. And on the test data, it gets 0.68 ROC curve. We can still see it's struggling with the precision and recall, but it's definitely doing better than the decision tree. So it's not the worst. Definitely could be better, but that's kind of that. So I'm not going to go too much further into optimizing these, but that is what you would have to do. You have to rinse and repeat. You go in, and there's many parameters. There's also ways of automatically learning good parameters that are going to improve your generalizability, but it's out of the scope for today. One thing we do want to do, though, is if we have a model that we think is doing pretty well, like maybe this one is okay. Another thing I need to mention is that even though it has all these, like this precision recall is very bad, we do have to remember that even though this 654 were marked as zeros, they're not actually necessarily wrong, right? These are wrong, the ones that it didn't get, but these are not necessarily wrong because they could just be novel genes, right? So that's another thing that we need to factor in that the plot isn't really showing us. Okay, so moving on, what we want to do is now that we have our model, we actually want to see what are the predictions. And so one thing you want to do is here we kind of shown that this generalizes, but we didn't give it all the data, right? There's some data it could still use to extrapolate further. So if we're kind of confident that our model can generalize, we can try training it on all the data to get kind of the most bang for our buck, use all of the data to make our predictions. We won't be able to benchmark these anymore, but we will get kind of the predictions with the most amount of data that we have access to. And so that's what this code does. It just trains on all the data, and then we extract the model predictions, and then we can show basically here is the probability that the model assigned, not to be confused with the probability of it being reality. This is just the model's own belief, which is definitely going to be biased. These were like the real results, so we can see this one it predicted to be true was actually true. This one it predicted to be true wasn't marked as true. So we can just filter out the ones that we already know, and then we have our novel predictions. So these particular genes, by priority, might be candidates for being implicated in eye diseases in this case, right? So we will want to look through these. In fact, I Googled a few of them, and I can say that just a few of them were in fact already, there's papers now about that they're implicated in eye diseases. Whether that, it's a qualitative assessment, it's a bit biased, but I couldn't find anything about this one, for instance.